This is my third users meeting and my first time talking. So thank you so much for the opportunity to tell you about the work that I do in the metagenomics group in Jeff Dangle's lab. I think Jeff's been collaborating with the JGI for about 5 years now, maybe, and it's been a really fun and productive collaboration. So I'm excited to tell you about some of the progress that we've made and some of our future directions. Okie doke. So we all work on root-associated microbial communities. So we look both in the soil surrounding the root system, or the rhizosphere, as well as those microbes present within the endophytic compartment, or the endosphere. So not only do we compare them to each other, we also compare them to soil where there is no microbe present. So on the next couple of slides, I'm going to show data that came out of a paper from Derek Lundberg, who was the graduate student who started the project before I arrived. So what we see here when we look at this first question of who is there in these communities, which is always your first question when you start looking at them, we can see that output here, we see that bulk soil and rhizosphere look very similar to each other in their phyla distribution, while the endophytic compartment looks much different in the distribution, as well as a decrease in diversity. So there are less microbes there, which is, of course, what you would expect. So for these studies, we performed just a 16S survey using 454 pyrosequencing here at JGI. And so what I'm going to show you with the immune system is how just 16S surveys have led us to sort of more sophisticated or directed questions, and the future services we're hoping to use here at JGI to start to tease apart those questions. So the major findings we found in the phyla distribution were that we see an increase in the endophytic compartment in phyla such as actinobacteria and to a lesser extent firmicutes, as well as a decrease in other phyla such as acidobacteria and proteobacteria. I have proteobacteria starred because at the phyla level, the distribution is smaller, although if you start looking at the family level, you can see that some family are actually increased in this endophytic compartment. And this includes things that you would expect like Rhizobacteriaceae, that sort of thing. Okay, so we wanted to ask the question, which OTUs drive these differences in the different sample fractions? So in this case, we've applied a generalized linear mixed model. And this is done by Suor, who is another graduate student in the lab. There's a whole row back there that's very heavily populated by the Dangle lab, and that's where you'll find most of these people if you have any more questions about the process. So Suor applied a generalized linear mixed model. And on this heat map, those OTUs which were found to contribute to differences are along the top. Along the side, you have all of the different samples. So blue indicates low abundance, red indicates high abundance. So you can start to see that there are different groups that emerge from this pattern. And a lot of these different patterns have to do with what type of soil the plants were grown in. So what your starting inocula is, or what your starting nutrient levels differs in what your microbial communities look like. What I want to focus on here are two groups. So first, over here on the right, we have a group of microbes which are highly present in the rhizosphere or outside of the soil and not very present inside of the soil or in the endophytes. And so this is true again and again between samples, between genotypes, between even soil types. So these are things that the plant tends not to want in its inside of its root system. The second group here, we also have a group of what we call our core endophyte, our core EC microbiome. So these are things that no matter what soil these plants were grown in, again and again, they seem to want them or require them. And this is where I pose my question. Because at this point, it appears that the plant is controlling this process because it's very consistent. And my question is, how does the plant immune system act to control this? So if a plant can't sense or respond properly to its microbial environment, how does it actually control this process? For this, I looked at 3 different groups of immune mutants, or 3 different immune processes. The first here is MAMP recognition. So the plant's recognition of things like flagella or elongation factor or peptidoglycan as a way to sense its microbial environment. This normally sets up a signaling cascade which turns on defense genes in a process called MAMP-triggered immunity. So when this process is disturbed, how does the plant still control its microbial environment? The second class that I looked at here is called pathogen effector recognition, and this is because plant pathogens have developed a way to inhibit this MAMP-triggered immunity. And it does this by secreting things called effectors through a type 3 secretion system, which then go over and inhibit this signaling process to turn off defense gene regulation. The plant has a defense response against this using a set of internal receptors, which recognize that its own plant genes have been, or plant proteins have been altered and turn on production of something called SA, or salicylic acid, which leads to something called hypersensitive response, or cell death, which is a localized cell death the plant uses to defend itself. And this process is called effector-triggered immunity. So salicylic acid is what's called a phytohormone. It's one of the defense responses we see. There are other phytohormones that we're investigating, such as jasmonic acid, or JA, and ethylene, which both lead to production of antimicrobial peptides, such as plant defensins. So when I look at my immune mutants, I look at two main classes that I'm going to show you about today, that I'm going to tell you about today. The first are hyperimmune plants. So these are plants that are constitutively making SA. These are plants that are always acting as if they've been infected by a pathogen. On the other side of that, we have hypo plants, which either don't make SA or don't respond properly to SA, or jasmonic acid and ethylene in addition. And those are my hypo-responsive plants. So I want to see what the difference is between just a wild-type plant, a plant that's always acting as if it's been infected by a pathogen, and one that can't properly respond to microbes in its environment. So here we have a principal coordinate analysis of weighted unifrac distances to try to get some idea what the composition of these things are. And what we see here is very similar to what we saw with Chris's data yesterday with the root microbiome, is that rhizosphere and soil create a much tighter spread than you see in the endophytic compartment. There's a lot more spread in the endophytes. My dream was that this spread would very clearly show the difference between the different classes, but actually what I see is that the different classes are still spread throughout here, so we're going to have to go another level deeper. So at this point, here we're looking at the phyla distribution between the rhizosphere and soil for both wild-type, hypo, and hyper plants, and you see that the phyla distribution is very similar. And the same is mostly true in the endophytic compartment, although there are some significant differences. So here we have what the wild-type phyla distribution is, and this is very similar to what we saw before. Actinobacteria is the highest. However, in both the hyper and the hypo immune plants, you have a significant decrease in this actinobacteria phyla. In the hypers, this decrease is partnered with an increase in proteobacteria, which we normally saw was down in the endophytes. And in the hypos, you see a significant increase in the firmicutes. So just to investigate this further, I'm going to break out these different immune classes into their genotypes. So there are 6 hypo genotypes, and we can see in the Bray-Curtis that they split out by their ability to make SA, which this is blind. We didn't plan for this to happen. This is just the way it came out. So those plants which don't make SA or make lower amounts of SA have an increased level of firmicutes, while those that make SA but simply don't have the downstream signaling have lower. So we have an opposite phenotype here, which is indicating to us that it's the level of SA itself and not necessarily the downstream signaling, since those plants that don't have the downstream signaling don't seem to control the level of firmicutes just fine. So that's the hypo story. On the opposite side of that, we have the hyper story. So when we break out the hyper genotypes, which ones do we see have this difference in the actinobacteria and proteobacteria? And when we look at those, we see that there's only one genotype, CPR5, which has this difference. The others look just like wild type. I should note that Natalie Brakefield, another postdoc in the lab, went through and actually did the root morphology on these things to make sure that the roots actually look the same. So they're the same in length, they're the same in lateral root numbers, they're the same in root hair formation. The roots look the same. There's something about the immune system in these plants that's altered that's driving this difference. So this difference between the other hypers and CPR5 could have to do with this signaling cascade. So it's known that CPR1, CPR6, and SNCC1 all use this PAD4-dependent pathway to upregulate the salicylic acid, whereas CPR5 does not use that. We're still in the process of determining the other parts of this mechanism to try to get at why it is that this one genotype is different from the others. But we do have this part of what we're trying to get at is what are the OTUs which contribute to these differences, maybe a clue for what the mechanism might be. So again, this was a model applied by Sur, the graduate student in the lab. In this case, instead of a generalized linear mixed model, we've used a zero inflated negative binomial. And this is just because our data set had so much sparsity in it. There were too many zeros to be able to decipher what was a meaningful zero from what was just background noise. So in this case, there are 43 different OTUs which contribute to differences between CPR5 and Columbia. And below, I've weighted them by their relative abundance. So here we have an example of a proteobacteria that's increased in CPR5. And one of the OTUs that contributes to this would be this pseudomonad. And then as an example of a CPR5 that's down, we have this actinobacteria, which is very much lower in the CPR5 compared to the Columbia. So in this case, the model is picking out some of those OTUs which were contributing to the differences we saw at the sequencing level. Okay, so just to wrap up the sequencing side of the story, we saw that there were significant differences both in the hyperimmune classes and the hypoimmune classes. And for both of those, that was manifested by a decrease in the actinobacteria. For the hypo, this was partnered with an increase in firmicutes, which we believe has to do with the level of salicylic acid itself and not the downstream signaling. Because those mutants which didn't make salicylic acid had increased, those which made it but didn't signal had decreased firmicute abundance. On the other side of that, in the hypos, we saw an increase in proteobacteria, which was specific to CPR5. Now, because all of these mutants are classified together as hypermutants because they all constitutively make salicylic acid, I propose that the CPR5 phenotype is salicylic acid independent, since it was the only one among all of the mutants which constitutively make salicylic acid which had that phenotype. But again, you have to test these things. So that's what I'm going to tell you about now. We've got some future directions. We want to confirm all of our phyla phenotypes using both quantitation via FISH, and we actually have this in the works already to repeat the sequencing with Illumina and using other V regions to make sure we're not seeing some sort of artifact. What I'm going to go into in a couple more slides here is a reconstruction community experiment we have to test these hypotheses. And then finally, we need to determine what it is about the CPR5 that makes them different from the other hypermutants. Okay, so the first question you always get when you're looking at these communities are what are the microbes present? So now we've defined that in two different studies. In Derek's study, which was published that summer, and then with this immune set. With the immune set, we were starting to get at how is it that the host controls it. So at this point, it looks like salicylic acid itself may be part of that control. I also want to say that while we were doing the DNA culture-independent the DNA sequencing of the 16S, we were also isolating microbes at the same time. And now we have a collection of about 400 different isolates we've been able to get out of the Arabidopsis roots, many of which are having their genome sequenced here at JGI. We also gave some of the root material to Scott Klingenpeel, who's been doing single-cell genomics, to try to get at more information about these communities so that we can ask those harder questions like, what are the function of these microbes? How do they interact with the plant? How do they interact with each other? And these are some of the questions that other members of Jeff's lab are trying to get at. So now I'm going to focus on how we're taking a collection of these isolates to do what we call a reconstruction synthetic community, where we take a wide diversity of things we've been able to isolate, reapply it to a plant, and see if we can ask more, and use that to ask more directed questions. So in this case, I'm using a collection of 40 different bacteria where we're adding them to the two sterile plants, and we're either adding exogenous SA or leaving them as they are. So we'll have both wild type, hypo, which don't make SA, and hypo that make SA but don't sense it. So an example of this is that within this set of 40 microbes, we have 6 of which are firmicutes. So we can ask the question, normally firmicutes are low in this group that don't make SA. If I now make SA, can I make that relative abundance go down? So we can start to ask those directed questions. And that's sort of where we are with that. So in this case, we'll do the harvesting to do relative abundances as well as other quantitation to investigate the differences between actinobacteria, proteobacteria, and firmicutes. And with that, I would like to thank you for your attention. I would like to thank the Dangle Lab, who's in that row back there, if you need to talk to any of them. Specifically, Natalie, Derek, Sur, Scott, And Jace, who is a special attendee because he's not here anymore, he's not at UNC anymore. Now he's at Berkeley, and he stopped over to say hi. So hi, Jace. And I'd like to thank all of our collaborators at JGI for helping us generate all of this data and everything. Thank you so much. Yeah.
So I've asked this of you previously, the direct assay stuff, and so there are some organisms. Have you been able to look at the genomes to see about salicylic acid degradation potential and what kind of pathways they might have?
So you did ask me this before, and I was hoping you were going to ask me again. Because I have taken that set of 40 strains to make sure that they can still grow in the presence of salicylic acid, and I'm starting to screen if they can use salicylic acid in degradation. We don't have the genomes back on all of these, but the ones we do have, we're looking for the degradation mechanisms for. Thanks for asking.
And so you've taken a very candidate gene approach to a genomic organism where they have knockouts of every gene. So have you thought about strategies to, more globally?
Yeah, so that was the original goal of Derek's project. When we did that initial survey, we took 8 natural accessions to try to use differences in genotype as a mappable phenotype. And what we saw is at that level of just different strains of Arabidopsis, it wasn't zoomed out enough to be able to see those differences. But we are very interested in that. And what we're getting at now is trying to stress it out and sort of drive it to try to make the phenotypes more extreme so that we can do that.